Hey guys, so there are some terms that I feel like I just know that are confusing you guys because some of you guys have talked to me about this and expressed some confusion over a few specific terms, mainly these guys, dihybrid and heterozygous, homologous. It gets kind of confusing because they describe different things that do slightly different things. They all describe genetic material, but different sizes and different shapes of genetic material essentially. So to break it down, I want to talk about first a couple different, we're going to start at the allele level, alleles and genes. And so I've drawn out these two DNA strands. One is the paternal strand and the other is the maternal strand. So if you were to turn these into chromosomes, they would look something like this, right? So here's your, here's your paternal chromosome, and then here's your maternal chromosome. Draw that in pink. And if you took each one of these strands, I mean, they're super long. This is only a very small section, right? We'll show in red that these are truncated, and to show that this is only a tiny section of a super long strand, and that this entire section is located in just this little tiny section of chromosomes, right? So remember, each one of these is a chromosome, and we're just talking about one of each here. So you have your paternal here and your maternal down low, all right? So let's remove some of the red. Don't want to confuse you guys too much. Okay, so let's talk about an allele. So we need to take a selection of each one of each strand. So your paternal chromosome here, let's say that we're talking about a gene that codes for height, and let's use a, sorry, I'm confusing myself here. So let's suppose the paternal copy has the dominant A allele for height, and then the maternal copy has the recessive A allele. So what that would look like on a DNA level, I'll show you. So if we're talking about the template strand, which ends up ultimately coding for a protein, let's give you a sample of DNA that would be the nucleotides that would code for this gene. So, and then we'll do the same thing here, AGCCTAGACAT. Not trying to communicate any subliminal messages here by the order of the base pairs, but so normally in a cell, you will have what will differentiate chromosomes from each other are the alleles in those chromosomes ultimately. If you have two homologous chromosomes, those homologs aren't going to be which would actually be these two. The paternal and maternal chromosomes will always be homologous to each other, but they will differ in a few base pairs, which ends up making different alleles. So let's say we change the A here to a G here, and then a C here to an A there. So we've just changed a couple of base pairs. That's just kind of an example, but in order to have different alleles, we have to change the base pairs in one of the strands. So let's say we change those two alleles, or change those two base pairs, and then that would make this allele an A allele. So here's the differentiation to make. We can have multiple alleles for a gene, but we can't have multiple genes for the same allele. Now what that means is, let's say this is the height allele, you can only have two spaces, or sorry, there are two kinds of alleles you can have for the same gene, for this gene, right? So for blood, there's actually the A allele, there's the B allele, there's the O allele, there's actually the RH alleles, what they call it, there's a D allele, there's actually a ton of different alleles for blood, to oversimplify, honestly. So we can have multiple alleles that fit into the same gene. Now, a gene is just a name for a section of DNA that codes for the same protein. So while the dominant gene here, let me backtrack a little bit. So while the dominant gene codes for one protein, here's a protein. This is pretty much exactly what they look like. Okay, this is the dominant A protein. The same gene, different allele, will also code for a protein, but it's going to look a little bit different just by changing even a couple nucleotides. So this is the little a protein. Right, and as we know, in normal dominance patterns, only the dominant A will get expressed, but as just an example, all we have to do is change a couple of nucleotides and it will change the ultimate protein that's produced, and that's what creates variation. And if this was a crazy different type of gene, maybe we would have a big A, a little a, and maybe an A superscript 1, A superscript 2 to indicate we have multiple alleles. So these are alleles, right? Remember, you can have multiple alleles for the same gene. Wow, can't spell. But you can't have multiple genes for the same allele. All right? All right, now that we've hopefully differentiated that pretty well, we can move on. So we want to review a little bit the terms heterozygous and homozygous. So homozygous refers to the genotype. So in this case, in this individual, they have a big A, little a genotype. The heterozygous and homozygous will be used to describe the different alleles that they have, the relationship that those alleles for the same gene have to each other. So remember, when we're talking about heterozygous and homozygous, we are only talking about one gene. On this chromosome, for example, there's a ton of genes. On any given chromosome, there are thousands or at least hundreds of genes. So we can't say that one full chromosome, so we can't say this chromosome is heterozygous or homozygous to this one, because that is way oversimplifying it. We're only supposed to be talking about one gene when we talk about homozygy. I don't know if that's a word. Homozygous and heterozygous. So heterozygous will only apply to one gene, but all of a sudden, and homozygous will only apply to one gene, right? If we have a big A, you know this. This is the easier stuff to remember. These are both homozygous genotypes, okay? Okay, but we've talked about something else, and that's dihybrid. We've talked about dihybrid crosses when we self-fertilize plants. We won't get into self-fertilization, how much that works. This video is more to describe just the difference between these terms. So a dihybrid cross is when you have two genes. So you have the A gene, which is, we'll say, the height gene. We'll type this out. And you have the B gene. I'm out of space. Let me make some space here. Okay, you have the A. I'll just type it all out. A equals height, and then your B gene equals, and I want to hyphenate this, and so just so we don't confuse. So the B alleles code for, let's say, eye color in humans. So in this case, we have two different genes, and we have two different kinds of alleles for each gene. And for this individual that we've drawn below, we have a big A, little a genotype for height, and for eye color, we have big B, little b. So if we have two different genes, and we have two different alleles, and we have a different allele in each space, this is a dihybrid. So basically the equation for a dihybrid cross is that you need two alleles, two different alleles, plus two different genes free, and that will make a dihybrid cross. Okay, so this is the meat and potatoes of of what a dihybrid cross is. That's all it is. It's just you have two genes and there's a different allele for each gene. Okay. I think that's pretty much it, and hopefully this clears up a few different issues that you guys have with this. I know it's complicated material. If you have any more questions, feel free to ask me. One more thing that I did want to go over is the difference between genotype and phenotype. All right, so okay, actually let's do something else. So let's add, as if this wasn't complicated enough, let's add another level of complexity to this. Let's say we're a triploid organism and we have 3 different chromosomes that code for the same set of genes. So we also have the A gene here, or yeah, the A gene there, and then the B gene here. But in a triploid organism, which we humans are not, we're not supposed to be, let's say this triploid organism has a 3rd chromosome with another A type and another B type. And let's actually make that a capital B just for fun. So what this would look like if you're phenotyping and genotyping this organism, let's just talk about the A gene. So originally we just had a big A, little a, right? There's the paternal gene, or the paternal allele, and then the maternal allele, and then we have a little big A, we have a little a, and a little a if we're talking about the triploid. So this is what a genotype would look depending on the ploidy of an organism, depending on how many copies of chromosomes that code for the same stuff that an organism has. So this is triploid, label that. Okay. Oh, I just got that backwards. All right, let's back up for a second. I keep doing this. So triploid, meaning you have space for 3 alleles, and then diploid, meaning you only have space for 2. Great handwriting. So this is what it would look like, and in triploid or tetraploid or heptaploid, whatever organisms, you wouldn't necessarily use the terminology paternal and maternal. We only use this naming system because we're diploid and it just is really easy and helps keep track of, with our type of reproduction patterns and how many chromosomes we have. So don't look too much into the paternal maternal chromosomes other than that it just helps us distinguish between homologous chromosomes. So this might, like again, repeating the same thing in different words, a triploid wouldn't have maternal and paternal, it would just be chromosome 3. I'll talk about that a little bit. I'll talk a little bit about that in the other video, excuse me, how to distinguish a little bit better. But note that each one of these is a different chromosome, right? You're going to have each allele on a different chromosome. And that you cannot have heterozygous or homozygous chromosomes. You can only have heterozygous or homozygous alleles or genes. Okay, so one more note on organisms of different ploidies. In diploids, these are totally different genotypes, right? Because you have different numbers of each, but the phenotype might be exactly the same. So same phenotype. But we have different genotype. Okay. So while on the outside, let's say we have a triploid human, while on the inside the cells would look really different if you had a triploid human. Assuming that a triploid human would survive, the phenotype would be the same because you have this dominant A. Now that's a different story if we had 3 A's, right? Because then you would have the recessive allele expressed instead of the dominant one. Okay, hopefully this clears up some things for you guys. Let me know if you have any more questions, and good luck on the test. Go crush it. Check out the next video, and we'll see you later.